the reverse transcriptase. Because we don't have, remember what's necessary for DNA synthesis to occur? We have the 5' phosphate, but we also have to have a 3' what? In order for DNA synthesis to keep going? We have to have a 3' hydroxyl. There's no hydroxyl there. So any reverse transcriptase reaction that uses this as a substrate can't keep going. It can attach it here, but it can't keep going. That chain stops. And so it breaks DNA synthesis from RNA. Not perfectly, but it really slows it down. Other viruses like Epstein-Barr, herpes virus 4, these guys can cause cancer in cells. Pap smears to detect abnormal cells. This is something that most of you are probably familiar with. What are we looking for here? We're looking for cells that are exhibiting behaviors, cell morphologies that aren't normal, which might mean that they are transitioning to being cancerous. If we can see them and identify them, then maybe we can get rid of them by just burning them away. Some of these viruses we can vaccinate for. That's what the Gardasil, the HPV vaccine. It's a vaccine not for all herpes simplex viruses, but for some of the ones that have the most risk of causing cancer. When it first came out, it was only suggested that women should be vaccinated with Gardasil. Now they're talking about, if they haven't actually already changed it, to say that young boys should also be vaccinated for Gardasil.
Women are carriers.
Right, for two reasons. One, there's a low chance that it can cause testicular or penile cancer. It's very, very low. But more to the point, protecting women is a good idea, but if you also prevent men from getting it, because their immune system recognizes it, then you can prevent that man from spreading it to any woman that he has sex with.
And there's no test for a man anyway.
Well, more to the point, because the symptoms are so low, there's no real test for it, because why would you spend the money looking for it? It's not going to hurt you if you're a guy, unless there's that exceptionally small chance it could cause testicular cancer for you, but who cares about that? So it's terribly tiny. It's not nearly the risk that women run for cervical cancer. Cancer can be caused by a lot of things. Here's the thing. Your cells have a lot of checkpoints. You are a multicellular organism that is highly complex and highly regulated. Remember, 97% of your genome isn't there to make proteins. It's there to control the making of proteins, right? So, what is cancer? A lot of your cells can be impacted by mutation that can change the sequence of DNA in your cells. And it might mean that that cell has been altered in some way that is semi-lethal. And your cells, most of them have checkpoints to check. And they can tell, you know what, I am so foobarred that I may as well just kill myself. And so your cells are constantly checking to make sure that they are still functional. And if they are not functional, they will commit seppuku. They will pull out the blade in and then up and then over and have the guy next to him checking to see if they'll scream. And if you're going to scream, then they'll cut our head off. And that's actually the way it works. But they lice themselves. They will commit suicide because they can recognize that they are no longer doing their job. And if this keeps up, they might become a threat to your body. But here's the thing. Mutations are random. Gold star, underlined, highlighted, boldface, in red, random. Which means sometimes the things that get changed are the enzymes that help the cell determine whether or not they're still doing their job. Sometimes the things that get changed are the enzymes that help the cell kill themselves when they know they're not doing their job. And if a cell stops doing its job, then that's cancer. Now, it might be benign cancer if it just stops doing its job and it's just a little mass of cells that aren't doing anything. But it could be very, very bad cancer if it not only stops doing its job, but then kicks on the part of cells where they think they just need to grow really fast. What happens to your brain if a few of the neurons in your brain stop being neurons and instead just start growing? Becoming bigger and bigger and bigger and bigger.
And your cranial pressure increases.
Cranial pressure is going to change. You're going to start pushing away and displacing all of the other neurons around them, even though they're still trying to do their job. And eventually it could put pressure on individual blood vessels that could seal off access to other areas of your brain, and you die because of a tumor. Aflatoxin, aniline dyes, arsenic, these guys can target specific organs of your body for mutations because of the biochemical processes that occur there that make them susceptible. How much danger do you think your bladder is from UV radiation? How much danger is your bladder in from mutation due to UV radiation? Not much. How much sun exposure does your bladder get when you're out at the beach? Not much.
Is it a nude beach?
Even at a nude beach, not much. Now, what about your skin? Much greater risk in your skin, because your skin's the thing that's absorbing all that UV radiation long before it reaches your bladder. There are a lot of things that can cause cancer. Chemicals, environmental exposure. Asbestos. You guys heard of that?
Stuff in the attic.
Yeah, stuff, if you have an old house, it might be the stuff in the attic. If it's ever detected, especially in some of these old buildings, if they're doing renovation, they have to come in and check to see if any of the material that was used in the floor, the ceiling, or in the insulation, or anywhere else for that matter, is asbestos. Because asbestos is quite literally just really thin fragments of glass. It's just like spider silk versions made out of glass. But lots of it. It works great as a fire retardant. It works great as an insulator. Problem is, if you breathe any of them in, those tiny little fragments of glass, they'll get embedded in your passageways in your lungs, and your lungs react very, very negatively to that. They will form cysts around it, and those cells will become irritated by it, and it will induce cancer. There was a decade or two there where we were using asbestos in a lot of stuff. Because it was the newest and neatest thing that we had developed in an industrialized society, until a lot of the people that worked with asbestos started dying of lung cancer. Not liver cancer, lung cancer. Because those little glass shards didn't make it to the liver, they stopped in the lungs, but that was bad enough.
We still use it.
It's still used to some degree, yes, but not so much as an insulator for construction and commercial housing.
It's used for kiln doors.
For what?
Kiln doors?
Kiln doors? Yeah, for heat transfer and absorbance?
No, because you can actually see through it when it's heated up.
Nice. We're just much more careful about it.
Well, it's a solid block of this stuff.
All right, who wants to see
Clone sheep.
a Cycloptean sheep?
I wouldn't mind seeing that video you mentioned Tuesday about from Harvard.
Oh, sure.
Several times before 11 a.m. tomorrow.
Baby.
Here, let's see if I can find the video.
There's a video tab right underneath the search bar.
No. Yeah.
Or you could go to YouTube.
Use a decent search on it.
Yeah, here we go.
Allah aşkına.
I screamed it.
Ya ağzına bak Asya'nın tek gözü var. Kulağın ufak. Sorgula bak. Ulan biri büyük. Oğlanmış bir de. Oğlan Oğlanmış da
Just in case you think that that's not real, that's like a puppet.
böyle oldu baksana sana. Erkek işte bu dün doğmuş akşamüstü. Bunun tek gözü var, kulağını bir ufak. Çeyresi yapıp.
It was a Middle Eastern country, I think. Well, let me ask you this question before I answer that one. What do you think's wrong with the sheep? You know, aside from the obvious, it has one really big eye and a weird tongue.
Missing a couple of chromosomes.
Missing a couple of chromosomes, genetic mutations. What else? Anybody else want to throw something out there? What do you think?
Well, we learned about viruses, so
Could be a virus, sure. Anybody else?
There's not much that's right with that.
Was that? Yeah, its ears are a little like this one's a lot larger on this side than that side.
And
Yeah.
his head is misshapen.
Yeah. Yeah, just right. The head in general is just all sorts of. Here's the thing: there's nothing wrong with that sheep.
Oh, okay.
Not a virus. It's not a chromosomal aberration. It's not even a mutation. That sheep's DNA is perfectly normal.
So that's what all sheep should
No,
No.
that sheep is all sorts of. But what happened is that sheep's mama, during a very specific 2-week period, I think between the ages of 8 and 10 weeks, got a hold of a flower and ate it. And in that flower was a little drug. And during that 2-week period of development, that drug screwed up the instructions that those cells used, that they followed in order to make a functional, normal, 2 eyes, regular ears, regular mouth, and everything else sheep. It'd be like me going, like you getting the instruction booklets for how to put together one of those really big swing sets or whatever, and I just went in with a really big eraser and just erased half of it and started drawing in with pencil or something. Just completely rearranged the way you use those instructions. There's nothing wrong with that sheep, except for the fact that the instructions in its DNA were not followed the way they should have been, because of a drug the mother was exposed to. Any other time during development, before that 2-week period or after that 2-week period, and it would have been born a perfectly normal sheep. But during that 2-week period, the enzymes that were expressed to control development at that stage of cranial development, of brain and skull development, that's when that drug can affect it, because those proteins are susceptible to that drug in that flower.
False hellebore.
Not before and not after. Did you find it?
Corn lily or false hellebore. It's Veratrum californium.
Right.
Californicum.
There's a common name for it. I don't remember the name.
Well, they just say it's also known as corn lily or false hellebore.
Corn lily, yes. And people who raise sheep for a living know what it looks like and know to get rid of it.
Or that or not.
Or that will happen. If the ewe eats it during that 2-week period, that happens. There is nothing wrong with that sheep. The reason I show you this is once again to impress upon you the importance of the 97% that has nothing to do with making a protein. That's what that drug does. It messes up how that other 97% works. Not by changing the sequence, not by deleting whole portions, not by getting rid of chromosomes. This is not a sheep with Down syndrome. Genetically, this sheep is perfectly normal. That's how important that other 97% is. Yes, the proteins are what make you you, but only if you make them in the right way, at the right time, in the right amount. Question?
It says the mistake of humans ingesting Veratrum californicum while pregnant is often due to hellebore, an unrelated plant with the same name, being recommended as a natural treatment for vomiting, cramps, and poor circulations.
Right. Because here's the thing, sheep are not the only ones susceptible. Everything from us to sharks is susceptible to cranial aberrations due to that chemical.
So if we eat it while we're pregnant.
At just the wrong time. Yeah.
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 End up with one eye.
Or something, yeah.
Does it have a mouth?
So those babies.
Yeah, it's
those
offset
faceless,
and
those
depressed.
babies are really-
Sit under its leg.
Yeah,
Does it have any eyes?
it's got the one right in the center.
Its tongue and mouth is like down here.
And of course, it's got a post-mortem incision.
Well, guess who's not eating that plant when I'm pregnant?
Don't go picking herbs and wildflowers in fields when you're pregnant, and you should be fine.
And be careful in potions class.
Whoa,
Nice.
Scared the dog.
All right.
It's a pretty good base for a...
Yeah, well, it's supposed to be the
There's got to be a subwoofer over here.
We don't actually need the audio for this very much.
They've got to be electrical.
All the cells are just attaching
Come on.
So is this the Harvard video?
Yeah.
to the cell. I think that's a better.
Oh, I was hoping it was going to be more like a Khan Academy type.
You just turn the volume down.
Yeah. You might be able to find a higher res version of it. Let's see. Let's see if this is better.
A lot worse because it's dropping frames.
Yeah, 720. There we go. It's still somebody's version of it though, because they've got the letters up there flipped. So this is the DNA double helix. Here is a enzyme coming along, grabbing it to help open it up. Or histone folding around it. So here's our DNA organization. So normally your DNA is wrapped around histones and slightly condensed.
I think it looks like it's a candidate for Tremors too. If you're even magnified.
Yeah, exactly. But then those helices are further wrapped and further wrapped. So this is DNA organization. We want to get to the part about replication. Where are we?
So is there an EQ attached? Because you're just lucky all your high end.
This makes me want to watch that Disney movie.
Now that, yes,
What
this
about the pill? Like the
The what?
The DNA, like they're cops, and then that pill, like the person.
Oh,
says
yeah.
Jones.
Oh,
Yeah.
Oh,
I don't know if you've ever seen it.
Yes, I'm familiar with what you're talking about.
I love that movie.
it's such an end movie too.
It's so underrated.
something Jones. He's a cop. I read about it like a couple days ago. He's what?
He's a white blood cell.
Oh,
Blood cell, but he's a cop white blood cell.
Yeah.
And then a superhero called the Pill comes in, and they end up attacking the virus together.
Here we go. That's what I'm looking for.
They don't get along at first, but then they make amends.
All right. So if you follow this, this is the DNA double helix coming in right here. There is the helicase is right here to open it up. So you have two strands. One of them is running out to this, running out all the way out to that DNA replication there, and the other one's running directly into that. This one is synthesizing the other strand continuously. So as this is split, it makes one continuous strand from one end of the chromosome all the way to the other if you gave it enough time. But the other one's running in the opposite direction, so it can't do it that way. So instead what it does is it breaks off these little sections called Okazaki fragments. So that's an Okazaki fragment kind of running around, and it'll start catching up until it gets to the end there. So here this extends out, but you know it's running down here, but this part's already synthesized. So it gets to that, it falls off, and then the next one starts running until it runs all the way down in that double-stranded. So this is just stringing out, and then it breaks off, and we're going to keep going until it gets... So the other one's going in the opposite direction, so instead of copying the whole length, it copies one little section, and then 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 other enzymes come in, and they will take where those two strands come in, so one side isn't complete, they'll come in and just seal that up again. So that's DNA replication involving a whole bunch of enzymes and two polymerases. Helicase to unwind it at the beginning, where it's coming in here, the double-stranded coming in. One strand goes down to this guy here, and the other strand goes out for Okazaki fragments for synthesis in the opposite direction.
Is there an increased risk of like, transcription errors on the
Nope.
Okay.
No, because it's still DNA polymerase doing exactly the same job. This DNA polymerase is the same as that DNA polymerase down there. It's just that it has to start over and over. It just stutters. And then something else comes in and seals in the gap and removes the RNA primers, which we haven't talked about. But DNA polymerase can't work from nothing. It has to have a 3' hydroxyl to start with. RNA can work from nothing. And so all of this starts with tiny little 10 nucleotides of RNA that are started as a starting point, and then that extends on to something else comes in later and removes the RNA, fills it in with DNA, and seals it all together. What is that? DNA transcription, just building the different enzyme complexes.
That also reminds me
Looks like it's
of the
flexible.
computer starting to use that.
And now if we do this,
Same thing over and over again.
another thing, Harvard life of the cell, inner life of the cell. This is fun. Definitely the highest res that we get on that. So that's the extracellular matrix outside of the cell membrane down the bottom. There's a cell membrane made out of lipids with lipid rafts.
lipid rafts?
Areas of the cell membrane that are denser phospholipids, and so they're sort of thicker and sort of frozen, floating around inside, so like icebergs floating around inside the liquid phospholipid. That's the inner layer of the membrane. Here's our cytoskeleton of a bunch of actin filaments that are cross-linked to give the cell its shape. All of these are proteins. Here's actin filaments polymerizing on their own to extend. Here is a secondary enzyme that comes along to destabilize the actin filament so it falls apart. Here's tubulin polymerizing itself to form the microtubules that your cells needs for the superhighways of the cell and also for cell division. It's what pushes it. Here we have a little myosin protein walking like Charlie Chaplin. He's carrying a tiny vesicle that is attached to it. Those proteins walk only one direction on those microtubules. That's a nucleus all the way down to the center with the mitochondria, little bacterial mitochondria squeezing itself through all those little tubes up there. Here's some RNA coming out of the nucleus with a protein cap attached based on its synthesis. Here comes a ribosome. There's the small subunit. There's the large subunit. And here we're making a protein. Conveniently enough, we only have one on each one. There's another mitochondria squeezing itself through like some sort of pupated worm. There we have a ribosome making your protein inside your endoplasmic reticulum. You can see little teeth marks from the RNA there. That's your Golgi or ER budding those vesicles that little guy is Charlie Chaplin walking along to deliver it. Those are vesicles fusing with the backside of the Golgi apparatus so that the proteins inside them can be modified and altered, and there's a vesicle fusing with the outer membrane so that the proteins that were made inside the ER modified in the Golgi can all come together in an organized lipid raft that will then activate them and extend out to grab onto the extracellular matrix to help adhere that cell into its environment. There's a leukocyte, white blood cell, deciding that it needs to invade the tissue around it and extending out like it's a amoeba and just kind of squeezing between two of the endothelial cells lining the edge of your red blood vessels. There you go.
You said bacterial, but
Mitochondria are complicit, facultative, mutualists. They are bacteria. They have a bacterial chromosome. They are bacteria that were swallowed by proto-eukaryotic cells 2.2 billion years ago, and they, instead of being eaten by those cells for whatever reason, they survived, cut a deal with them, and so now they are the powerhouses of the cell. That's where all of your aerobic respiration happens. So without the mitochondria, we wouldn't be able to use aerobic respiration. We wouldn't be here because multicellular life requires aerobic respiration. It costs too much energy. So they're there, but they are the descendants of bacteria from millions and millions and millions of years ago, and now they are so specialized that we cannot grow them outside of cells. If you pull them out of a cell, they'll die, because half of the proteins that they need, over half, aren't even in their own chromosome anymore. They've been shuttled to the nuclear chromosome of the organism that they live in, but they replicate their own chromosome just like bacteria do. They go through binary fission to increase their number just like bacteria do. And they use exactly the same ATP synthase that bacteria do to make ATP. They just do it in the microenvironment of the cell. Chloroplasts, the things that make sugars out of CO2 and sunlight, those are also mutualistic bacteria that have their own bacterial chromosome that are responsible for their own replication and most of their own biochemistry. And those are not the only two. There are many other examples from other lines that have absorbed other prokaryotes, and those prokaryotes have become mutualistic organisms within them, but for whatever reason, those lines didn't take off the same way eukaryotes did or Plantae did as an offshoot of eukarya.
How is that on 43 and the first I've heard bacteria associated with chloroplasts or mitochondria?
Because it's a detail that your average fourth grader doesn't need to know.
Yeah, but AP Bio?
Yeah, you'd think that they would have mentioned it. All right, so that's way outside the box. So I'm going to go turn on the light. Did you pull up the question on the homework?
I did.
Did you figure it out?
Well, at that part, it looked like circles to me, but after, like
Looking
on
at
the
it.
review, it looked like check boxes.
So the confusion was not so much which thing did or did not belong, it was there's more than one correct answer, and that
Yeah.
confused you? Yes, I
So
can
on
understand.
the first, like, I
Oh.
clicked one, and I was like, why is this wrong? And I clicked another one, and like, this is wrong again.
Wrong again!
So, yeah, it would be
Yeah,
kind of
that's
helpful to
fine.
have like all that apply on there. Just saying.
Just to preface the questions to make sure click all that apply in the instructions.
For the
Yeah,
ones who can't
I could
tell
see
the
why that would
circles.
be useful. Yeah, I could see how that could be helpful. All right, so here's the deal. There is a column in your grade book. I don't want to open it right now to show you because I would be giving away someone's grade.
I don't care.
There is a column in your grade book that is labeled, what is it labeled?
Minus
It
D
is
for
labeled, there's actually two columns that I want to point out. One of them, just making sure that's off, is homework total. So, or I guess in the grade book it's little t underscore HW. So that's your homework grade out of 100. That 100 is the equivalent of a test grade, right? So you can ignore all of your other individual homework assignments and just treat that homework as a 100-point exam, just like the exams are 100-point exams based on their percentage, right? So that homework total and your current total for exam 1, exam 2, and exam 3 are added into the C total column that's near the end. That gives you your current running tabulation. That is how many points you have right now in this class. So, if you were to look at the syllabus, it says that the grade for an A cuts off at 425 out of 500 points. 100 points from homework, 400 points from 4 exams. So, you can find out what grade you need on this last exam by subtracting your current total from 425. If that leads you with 73, then you need a 73 on exam 4 to get an A. If it leads you with 170, you aren't going to get an A.
How many do you need for the?
425 for an A, 350 for a B. It's in the syllabus. So the syllabus tells you where the cutoffs are for the individual grades. What I can tell you is the majority of you are within spitting distance of a B. which means the majority of you are within a very reachable distance of an A. Some of you a little bit more so than others. So if you want to figure out how you need to do on this exam, which opens when? Tomorrow at 8 a.m. and closes when? Sunday at 11:59. And unlike the previous 3 exams, on this one there is no makeup. If you don't take it by then, you get a 0. And whatever grade you have currently will be the grade I give you. Right? So, don't forget this weekend. I would love to give you some more time, just in case there's a mistake, but I can't. I have to have grades turned in, but I just can't do it. So, by Sunday, that is the cutoff. Sunday at midnight. And it'll tell you the grade right off the bat after you've done it. I changed the view so that as soon as you're done, you can go in and tell what your grade is. It'll also show you what questions you have. It won't give you what your answer was or what the correct answer was, but it will, if you want, it will tell you if you got it right or wrong. So you can go in and see which topics you missed if you care. So that's already enabled. But that's how that's going to work. So on Monday, I'm going to open up that grade book, and anyone that has 425 points and up, I'm going to give you an A.
When's the test due?
Sunday at midnight,
Okay.
just like the previous 3. So I'll wait until Sunday after midnight, or more likely Monday morning when I get up, to start the calculations. Now, I believe there is actually there is a final adjusted grade and final calculated grade column that currently has nothing in it. When I do that, those should be populated, just like any other class that you're looking at in D2L, and so you can see what I gave you. Hopefully there won't be any, just discrepancies between that. If you got 212 points and I gave you an A, then I did something wrong. If that's the case, you might not want to point it out. If you have 487 points and I gave you a D, you should definitely point that out. So, that type of thing. Question?
I just have a question about how the homework was calculated.
Yes. In the syllabus it says if you finish all 7 of the homework assignments, then you get the 16 points that are left out of 100 for free. So it was a incentive to do all of the homeworks. So we have 7 homework assignments worth 12 points each. That's not quite 100 points, and so to make up that 100 points, I just padded it with what would have been the 8th homework assignment, sort of. But since this is my first time teaching it, perhaps I didn't impress upon everyone the importance early enough in the semester, I decided just to say screw it and give everybody those 16 points anyway. So that homework grade is including those, so it's 16 plus whatever you got in all your homeworks, that's where that number comes from.
Gotcha.
So,
So we already have that in our...
it's already in there, it's baked in. So quite literally, take the grade you want, subtract the points you have, and that tells you what you need on this test. So if you want to get a B, if that's all you care about, if a B is good enough, most of you are within, let's just say an F on this last exam, and you'll have your B. So, but that means that if you do well on this exam, you could have an A, right? Now, if you need 130 points to get that A, then you're not going to get an A, because there's only 100 points left. Even if you got a perfect on this exam, you're not going to get 130 points out of 100. But it does mean that, you're pretty close to getting a B if you do okay, right? So, I do not encourage anyone to slack off on this exam, because the last thing you want to do is say, I only need 47 points, I'm good, and then not really try and only get 46.
Right.
Oops.
I've got that A now.
Right, so, because that's the thing. I try to be as nice as I can, I try and be as accommodating as I can, but I feel like I give you guys a lot of opportunities and advantages, shall we say, in the way that the grading system works, the way the openness of the exams and everything else. So if you're a point shy, you're a point shy. If you'd have tried a little bit harder at upwards of 16 other points earlier in the semester, you wouldn't be 1 point shy, right? So I don't feel so bad. I would love for all of you to get A's, as long as all of you actually earned it. That's the way it works. It's hard to flunk one of my courses. You really gotta try to flunk. But in my history of teaching classes, I have had a handful of students who have succeeded at flunking. And so I gave them the F that they evidently really wanted. So hopefully that is not any of you guys. So that's the way grades work. That's the way the test is going to work. Does anybody have any questions about anything else? 14, 15, and no, 15, 16, and 17. 3 chapters, which means there's going to be 16 questions each. So it'll be the same length as all the others. We'll just have more questions per chapter. Still going to be random, still going to be multiple choice, true, false, just like before. I think that about covers it.
Okay.
Anything else? Then thanks for coming, guys. I hope you learned something. I truly do. But that's all I got. And I wish you a good luck.
How do you have the exam calculated?
What's that?
How do you have the exam?
Oh, it's the percentage. So not like 42 out of 48, it's the percent that matters. That's a good question.
I was
So
like,
in-
because the
Yeah, in the future I'm gonna have to change it.
that's so confusing.
Right, no, don't use any of that. Look at the total, look at the current total. So it's the percent. If you got a 78%,
So worry about
It's
this
78
right
points.
here.
worry about, about that. Those numbers there.
Okay, so this is calculating.
Correct.
I was like, so confused.
Right, well, and that's the thing is that I don't like this, like points, but your point calculated total, yeah, so you got those 4 things plus the total homework. So that's your total homework.
So this is your calculating grade.
Correct. Yeah, it's adding your percentages here, plus percentages, plus percentage, plus percentage, and it's giving you that many points.
Okay, so that's points, not percent.
That is percent of that grade. You currently have that out of 500, but that's not counting
Okay.
this grade here.
Seems weird.
Well, I can't make it do it differently.
Yeah.
So yeah, it's like, it uses these percentages to add up points right here. And then that gives you your grade out of 500. So here's 100 possible points, 100 possible points, 100